the in vitro guidance, the main purpose is to inform the need of in vivo studies and define the potential for drug interaction for a new drug, either as a substrate or as a perpetrator. So first of all, I will talk about the interaction mediated by enzyme. There are the major enzymes are listed here. It's 1A2, 2B6, 2C8, 2C9, 2C19, 2D6, 3A, and this is the same as 2012 guidance. It's a kind of a routine to evaluate a substrate as a CYP substrate because most of the drug metabolized by CYP enzyme, and we want to know how the body, eliminate, get rid of a drug. And also, it's a kind of routine to evaluate a drug as a perpetrator, as an inhibitor or inducer of a CYP enzyme. And then we will apply some cutoff values to determine whether there is a potential for the in vivo interaction. As I will mention here, there's called the R value, and we compare this R value with the proposed cutoff of the threshold. And if it's above that, then it means there's a potential. If it's less than that, it's unlikely for a drug to interact with a CYP enzyme. So there are two major mechanisms. One is a reversible inhibition. The other is a time-dependent inhibition. So for reversible inhibition, there's a R1 value is calculated by 1 plus Imax unbound divided by Ki and compared to a cutoff value 1.02. So this Imax is the steady state Cmax of a new drug as inhibitor in plasma at a therapeutic dose. And the U means the unbound or the free concentration. So the Imax U is calculated the product of Imax and FUP. And this Ki is an unbound inhibition constant determined from in vitro experiment. And for CYP3A, because it's also present with a substantial amount in the intestine, so for evaluate the potential for DDI, we also need to consider the concentration in the gut, so that will be the I1 gut. Is calculated by IGUT divided by Ki, and this IGUT is the dose divided by 250 mil. This is a conservative estimate and likely to be higher than the real value. And the time-dependent inhibition equation is listed here, but I won't go into detail. So what's the difference from the 2012 guidance? I mean, first of all, we use the inhibitor concentration, the free concentration, because it's usually considered that it's the free concentration of a drug to interact with the enzyme, receptor, or target. And the threshold is changed from 0.1 to 0.02. This is the same as EMA. And as you know, that Imax unbound divided by Ki over 0.02 essentially is the same as R1 value of 1.02. And for the TDI, we also align with the EMA criteria by multiply a factor of 50 with the Imax unbound. And this is a sort of arbitrary factor. The purpose, the intention is that when you use the unbound Imax, it will decrease the concentration compared to the previous one, which is the total concentration, and it may cause some more false negative. So to balance that, they introduce an arbitrary factor. And the EMA, I think it also suggests calculated the potential for TDI using the gut concentration, but our guidance doesn't include it. It's because the TDI is well known that the prediction usually is over prediction. So if you introduce further the IGUT, it won't. It will just increase more, or will make the same false positive prediction. So what's the rationale for this change? And the reference is listed here. Around 2014 or 2013, our colleague conducted a comprehensive evaluation based on a number of DDI in vivo studies conducted with midazolam. That's a probe substrate of CYP3A. And then they evaluate the performance of different criteria, including the FDA criteria in 2012 guidance and also the EMA. And based on the evaluation, we found that the performance in general is similar between the two agencies' proposed criteria. So to help harmonize, we align with the EMA for some of the criteria. And this is determine if a new molecular entity is inducer of CYP enzyme. This is basically the same as the 2012 guidance, so I won't go into further. Just to mention that PGP can also be induced And in general, it's less sensitive than CYP3A. So if the drug is not inducer of CYP3A, then we usually think that it won't induce PGP. And recently, there are some literature findings suggest that OATP could be also induced by some drug, but that's quite a new area, and maybe we will see more results. So for the evaluate the induction potential, there are several methods. One is the fold change method. This is focused on the change of mRNA. For example, if the increase is at least twofold with your drug, and a response, the increase is at least 20% of relative to the positive control, then there's a potential for the drug to induce the CYP enzyme in vivo. And we also provided the option for a more more advanced method called the correlation method or basic kinetic model. For those two, you need to characterize the Emax EC50 from an in vitro experiment. It's just like the general pharmacological experiment. Determine the Emax EC50. And compared to the previous guidance, we add back the activity. Previously the 2012 guidance, we primarily focus on the mRNA change. But just remember, there's some limitation with the activity assay. One is that if your drug is an inhibitor of the CYP enzyme besides induction, then this activity will kind of become complicated. They're confounded by the inhibition effect. The other thing is that the activity, usually the range of change is less than mRNA, so that may cause some difficulty in determining the Emax EC50. So as a summary, for determining a drug as a perpetrator enzyme, one is an inhibitor, the other is inducer. The inducer more focused on 1A2, 2B6, 3A. And then we determine the in vitro parameters based on the basic model which I described. We determine whether there's a need for in vivo study. However, it doesn't mean that if the value above the cutoff, then definitely you have to go for in vivo study. We also provide the option for using other approach like the mechanistic model, static or dynamic. Dynamic usually means that PBPK. So you can use that to justify if the drug has a potential or not. If this suggests that the drug may not have a significant effect in vivo, then you don't need to conduct an in vivo study. Now I will switch the gear to transporter, as a substrate or as an inhibitor. As Kelly mentioned, there's as a substrate, it's mainly the three-group transporter. One is the PGP BCRP. As you know, it's an efflux transporter present in multiple organs. It's limited drug absorption in intestine or mediated biliary secretion in liver or renal secretion. And it may also limit the penetration of drug into brain because it's present on the blood-brain barrier. And the other group is uptake transporter called OATP 1B1, 1B3, present in the liver, mediated uptake of a number of drugs into liver. And the third group is a renal transporter, the uptake transporter OAT1, OAT3 for ion drugs, OCT2 is for cation drug, MATE is the efflux transporter on the lumen side of the tubular cells, which mediate drug excreted from a kidney cell into urine. I just want to mention that for this renal transporter, it's needed only when the active renal secretion is at least 25% of the systemic clearance. Lots of the time you will see some drug eliminated from urine, but that's mainly accounted by the glomerular filtration. So if you inhibit the transporter, you still won't affect that part, and the renal elimination largely remain similar. So you don't need to worry about the drug potential. And on the other side, it's a kind of more routine to evaluate, investigate a new drug as inhibitor of a transporter. It's because a drug not being a substrate of a transporter does not necessarily mean it won't be an inhibitor of transporter. And then we will apply the cutoff value, apply certain criteria, and compare to the cutoff value, just similar to the CYP enzyme. And for the induction, as I mentioned, that it's not well established, but it doesn't necessarily mean that you don't need to worry about it at all. So this is the comparison of the 2012 guidance and the current guidance. For the PGP, we kind of simplify the criteria from 2 into 1. Previously, we consider I2 and I1. I2 is the same as the I got. That's a dose divided by 250 ml. And this I1 is the total Cmax that represents the inhibitor concentration in plasma. And now we only mention that the I got. In the OATP 1B1, 1B2, we simplified from 2 steps into 1 step. In the OAT1, OAT3, it remains the same. And for the OCT2 mates, the change is that we added the mate because there's a large overlap for the substrate and also inhibitor spectrum for OCT2 and the mates. Actually, a number of drugs, they have a stronger potential to inhibit mate than OCT2. So the interaction mediated by OCT2 we used to think about actually probably is really mediated by the inhibition of mate. And also we updated the criteria for that. If the ratio is above that, then we consider that there may be potential for the in vivo inhibition. Here is just to give you a quick shot of how this criteria is updated, and this is about PGP. I have the slide for OATP 1B1 and the renal transport in the backup. So my colleague in the antiviral team, they conducted some research based on a number of studies, 50 or 60 in vivo studies conducted with digoxin as a probe substrate of PGP. And then they compared the prediction performance of the EMA, FDA, and different criteria. And they found that using the I2 alone, that's mainly about the intestinal PGP-mediated interaction. Actually, the performance, false negative, false positive, are quite similar to the other criteria. So that's the basis why we simplified the criteria. For the drugs which are given by IV or the inhibition moiety actually is the metabolite, for those, this is not really applicable because this is I2 is the gut concentration, and in those cases, the inhibition actually happen at the systemic level. So for those, we still need to further evaluate the data to define the optimal criteria. So now I will switch to give a case example, dolutegravir, as many of you are quite familiar with that. And this is a real case. Dolutegravir is already administered. The Cmax here is 4 microgram per ml at a 50 milligram BID dose, and it is a highly protein bound, close to 99%. So the sponsor conducted an in vitro study, found that it is an inhibitor of OCT2, and they characterized the IC50 close to 2 micromolar. So the question here is that does dolutegravir inhibit OCT2 in vivo, and is it necessary to conduct an in vivo study to evaluate the effect of dolutegravir on the PK of OCT2 substrate, for example, metformin? And so we calculated, and so if you calculate the ratio of unbound Cmax divided by the IC50, you will get a ratio of around 0.05. So that's less than the cutoff value we propose, 0.1. So then from this, you will conclude that dolutegravir won't inhibit OCT2 in vivo, and you may not need to consider further in vivo studies. However, a little bit later on, there's a literature published in Kidney International Journal. This is from Gilead people, dolutegravir is from GSK. And they found quite different results. So if you look at this table at the bottom, dolutegravir actually inhibit OCT2, which are much lower IC50, that's 0.066 micromolar. So that's about 20 to 30-fold less than the IC50 determined from GSK. So the lower the value is, the more potent the inhibitor is. So, and this is further confirmed by some later study conducted by Merck scientists, which also reported IC50 of 0.21 micromolar. So if you use this new value, for example, here is 0.11 micromolar, conducted in vitro when you use metformin as a substrate, you will get a ratio of 0.89. So that's much higher than the 0.1 cutoff. And from this, we will expect that there will be in vivo interaction. So what's the true effect? And as some of you may know that actually dolutegravir increased metformin PK AUC significantly. And this is the PK profile of metformin. The lower two are the metformin given alone, either before or after dolutegravir co-administration, and the upper line is when the two drugs are co-administered. Actually, dolutegravir at a 50 milligram BID dose, it increased the metformin AUC to 2.5-fold. And because of that, in the labeling of dolutegravir, we recommend limited the maximum dose of metformin to 1,000 milligram. That's because, in general, the maximum dose of metformin is about 2,400 milligram. So, divided by 2.5 is exactly 1,000 milligram. And also, you said that the labeling recommend contraindicated with dofetilide. That's the OCT2 substrate with narrow therapeutic index because of the QT risk. Because we anticipate that dolutegravir may also increase the concentration of dofetilide significantly since they share the same pathway, the OCT2. So what's the reason for this difference of the in vitro data from the different stakeholders? And here is a slide I borrowed from Dr. Yun Huang from Optivia. They further looked into this, and they found that the reason probably is because non-specific binding in the in vitro experiment. So here they also use metformin as a substrate so that they will have a common ground to compare the results. And in this figure, as you see, the right one is used the nominal concentration. It's like if you added 10 micromolar for dolutegravir into the plate, and you just use this 10 micromolar. However, if you measure the concentration, you will find that the concentration in the preparation actually is lower than 10 micromolar. That's because quite an amount of dolutegravir stick to the wall of the plate. So the concentration in the medium actually is significantly less than 10 micromolar. If you use that measured concentration, you will find that this curve shifted to the left-hand side, so that the IC50 you determined is much less than the one compared to the one you use the nominal concentration. So the concentration, if they use a measured concentration, actually it's get close to what reported by the Gilead scientists. So this is just to give you an example that we need to pay attention to how this in vitro data are generated rather than just plug the number into the equation and calculate the ratio compared to the cutoff and then make the decision. So as a conclusion, the in vitro experiment results, it will inform the need and the timing of in vivo study. We want to leverage the knowledge and the scientific understanding about the system to determine the in vivo DDI potential using an integrated approach, like I use the simple model, as I mentioned in the previous slide, and the mechanistic models, which I didn't talk about due to the time limit, but you can find that in the guidance. There's a table or figure listed the details, and also the dynamic model, which is PBPK. And the criteria of this simple model, I want to emphasize that it continues to be updated, especially for transporter, because it's a quickly evolving area. So like the matrix, the cutoff value, this will be updated when more data become available, especially like for OC2 and MACE, which we have relatively limited data, and with the new data generated, the criteria could be updated. And finally, as an example, I highlighted by the example, it's important to standardize and validate the in vitro assay conditions to ensure that the in vitro data generated has good quality. For example, the proper controls and check the recovery or mass balance from the study and pay attention to the potential non-specific binding. And this non-specific binding not necessarily happen with all the drugs with a high protein bound, but a highly protein bound drug have a higher tendency to have that. So finally, I want to acknowledge a number of my colleagues, my current or my previous colleague. They contributed a lot to this new guidance, especially Dr. Lei Zhang and Dr. Kelly Reynolds. I borrowed a lot of the slides from their previous presentation. And finally, thank you very much for your attention.